Our next talk was going to be Tony Wyss-Coray, but he couldn't come today. So instead, he sent Hamilton Ohm, a PhD student in his lab, who has a lot of energy and enthusiasm, and he's going to tell us about how we all are aging. Hamilton.
Thank you. Hello? Mic check? We're good? Okay. Wow, hi. Hey everybody, I'm Hamilton, PhD student in Tony's lab. First of all, I want to say thank you, Larry, for the invitation. It's honestly such an incredible meeting that you've put together. Things I really don't think about often, like chronic pain, depression, stress, trauma, socioeconomic, racial factors that all have such huge impacts on health that I think are honestly probably way more important than the small molecular things that we're trying to drug. So my talk's more on the molecular angle of how we age and trying to measure that and slow it down. But I think I really do not want to take away from the previous talks. I think that we can do so much for our health by just being better human beings and treating each other better. So with that, I'll speak about the molecular biology of human aging. Okay, so I'm interested in this question of how we age and how we could potentially slow aging and prevent disease, because really age is the strongest leading risk factor for so many diseases, including heart disease, dementia, and as earlier speakers have alluded to, we've sort of in medicine treated these diseases individually as separate entities, but really they're all connected, and what connects them is aging. And so by slowing down the aging process, can we potentially prevent disease, stay healthy for longer, and live better, high-quality lives. And so there's been a lot of promise and excitement in the field of being able to understand what aging is at a molecular level and also treating it based on studies from model organisms. So all animals age, and actually in pretty conserved ways. Some conserved mechanisms that I'll explore a little bit, but we study mice, even worms and flies and dogs and monkeys to try and tell us about how animals age and hopefully translate that. And so some of the key findings are that there are these conserved pathways in aging related to growth signaling, stress, DNA repair, and studies show that you can actually intervene on these pathways to extend the health span and lifespan of model organisms such as mice here using rapamycin, which is an mTOR inhibitor of a growth signaling pathway, but also you might know of caloric restriction, so intermittent fasting, those kinds of things, pretty reproducibly extend health span and lifespan in model organisms. But how do we translate these findings to humans? Will these drugs even work in humans? I think that was one of the key questions that I was interested in coming into Tony's lab, is to really study human biology. And because we're pretty different. 80% of lab mice actually die from cancer. Humans die from many other things. We're also, as we know, much more complex social beings and so many more impactors on human health. And so I wanted to try and find a way to understand aging at the molecular level in humans. And so the way we came about that was to probe the blood. And the reason is that the blood connects basically every cell and tissue in the body. And you can actually sample molecules from the blood to understand and get even diagnose patients on the status of their certain cells and organs. And for example, when you go to the doctor, you typically measure blood cholesterol to see, is your cholesterol high or your arteries clogging up? We measure many other things, including enzymes in the liver to assess liver damage. But really with the advent of new technologies in proteomics in particular, led by SomaLogic, co-founded by Larry, of course, and then also Olink Technologies, we're now able to probe thousands of proteins in the blood pretty reproducibly across tens of thousands of samples, of human samples. And so really we have unprecedented scale of acquiring molecular information about human biology. And so we sought to utilize this information to try and understand something about the aging process. And so this was a study in 2019 led by a postdoc named Benoit Lehallière at the time, who looked in a cohort of roughly 4,300 people. So what they did was they measured the levels of roughly 3,000 proteins across 4,300 people across the lifespan, so people aged 20 years old all the way to 100, and just asked a simple question: which proteins in the blood change with normal aging? And what he found was that actually there's really quite dramatic changes with age. So the blue is low, black medium, yellow being high. So you can see a lot of proteins here that seem to really increase with age, and actually sort of interesting in that they increased really dramatically in the later decades of life. So complex process, nonlinear process, and also some proteins that also decrease with age. And if you do pathway enrichment on these proteins to try and understand what these proteins may be doing, you find some proteins related to axon guidance, so proteins involved in your nervous system, blood microparticles, many other different pathways, including extracellular matrix, many that are relevant in aging. And so this was a really encouraging study to show that there's actually a lot of things that change in the blood with age, and maybe the levels can tell us something about the aging process and the person's risk for disease. And so one of the cool techniques that you can do with modern statistical techniques and machine learning models is to get an estimate of one's biological age based on all of their protein levels. And so what that means here is it's basically you train a machine learning model to predict an individual's age based on their protein levels, and so you end up with a graph that looks like this. And so each person has their predicted age and then their actual age. And so you can see some people really deviate quite dramatically from their actual age. And so this person in the red is a lot older. Then they appear a lot older based on their proteomic profile, and this person down here appears a lot younger. And so we call that having a positive and a negative age gap. So that'll be an important terminology that I'll continue to use. But I think a really intuitive way to think about this is Obama before and after presidency. It's like chronic stress, depression, not really really bad for you. This guy right there, that was me after my PhD, after 25 years. No, I'm kidding. My PhD was really fun. Okay, so that was a study in 2019 which used basically thousands of proteins in the blood to try and get an estimate of each person's biological age. But the key insight that we came into with this new study was that human aging is really quite complicated and cannot be summarized by a single value. And it's really quite obvious if you look at what people die from. People die from all sorts of things, mostly heart disease in humans, but also cancers, of course, but then chronic respiratory diseases, digestive diseases, neurological diseases, and so many others, which suggests that even within the same person, our organs are aging at different rates, and that probably varies across the population. And so we really wanted to try and understand and measure aging at organ resolution. And I think this is going to be increasingly important as medicine advances, because we can no longer rely on like a single readout of aging, which we typically associate with mortality risk. Just because we extend someone's lifespan doesn't mean they're living healthier. People survive heart attacks now, people survive a lot of different things. And so if we can prevent dementia, if we can prevent heart attacks by understanding brain aging, heart aging, I think that's going to be really key. So that was the idea, to try and understand aging at organ resolution. And the way we thought of doing this was to, instead of looking at all of the proteins in the blood to try and understand aging, we just look at the ones that are coming from specific organs. So actually, when you go to the doctor, you measure enzymes that come from the liver to assess liver damage. And so in the same way, can we identify proteins at scale, which of them come from the kidney versus the lung versus the brain and the heart, and can we derive these estimates of organ-specific biological age using those proteins? So that was the key hypothesis, and so this was a study co-led by me and Jared, a former grad student in the lab, and we published this recently. So just to give an overview, so we first tried to map the plasma proteome, the origins of the plasma proteome. Which proteins in the blood, out of those thousands of proteins, can we confidently say come from certain organs? And the way we did this was we integrated the proteomics data with human tissue bulk RNA-seq data. So here's an example of a gene that, based on the expression levels of that gene across various tissues, we know that it's very highly expressed in the brain, very lowly expressed everywhere else. So we call that a brain-specific gene. What's cool is that you can find this gene, or the protein encoded by that gene in the blood. And so then we can sort of confidently infer that that protein is likely coming from the brain. And we do this across all the proteins in the platform. And you can find brain proteins, kidney proteins, heart proteins. And so this is the sort of distribution that you get. Roughly 20% of the plasma proteome seems to be derived from a single organ, many of which come from the brain, which was quite surprising at first, because I think you usually think of liver proteins as being the dominant proteins in the blood, which is true by concentration, but actually by the unique number of proteins that you can measure, there's lots of them coming from the brain. And so this was really encouraging for us to maybe we can actually understand brain aging. And so we used these organ-specific proteins that we identified to train organ aging models. Sorry, this graphic isn't there, but you can imagine. Ooh, I can use my colors. So it's supposed to look like this, where there's a lot of people here who look older for their age. Can I change colors? No. You can imagine this is blue. A lot of people here who look younger for their age. And so we can have these sorts of biological age estimates for each organ to try and measure aging at organ resolution. One of the key things that we needed to determine was, are these different biological age estimates actually giving us distinct information about that organ? And so the first thing we did was try and just get a general overview of the correlation between different organ age estimates, or the age gaps, so how much older or younger one appears. And we found an interesting pattern. So there, even within a person, one could have pretty drastically different age gaps. And here's sort of an example where we compare 3 individuals who have, I should mention, we also trained a conventional aging model, which uses all of the plasma proteins to get a biological age estimate. So here's three people who have a conventional age gap of 0. So they are basically population average based on a single model. But when you look at their organ aging profiles, they're really drastically different. So the person on the left has broadly youthful organs, the person in the middle, some young, some old, the person on the right, not looking too good. And so this suggests that each estimate is telling us something different about the biology of that person. And broadly, if you look at the correlation between any 2 pair of organs, they're generally, oh man, time to draw again. Oops. This is so fun. It looks like this. So overall, there is some degree of correlation between age gaps, which makes sense because the whole body's connected. But on average, the correlation is roughly a Pearson correlation of 0.25, so mild correlation at best. which suggests that each organ age estimate is really telling us something different. So we also identified these types of extreme agers, and I'll skip to this one. But basically people that have just one organ that is particularly aged compared to the rest. And this was really quite striking. We sort of thought, I think, that aging is this organismal process that happens everywhere across our bodies. But in a few subset of people, so this is like 2% of the population, they appear to be aging really quite faster in a single organ. And actually many people, or another 2%, that have aged organs across their whole body. And so this is really quite interesting and suggested that really in humans as well, organs age at different rates. And we can begin to try and measure this. One of the key questions still, though, was, does being older or having an older biological age estimate actually mean that your body is physiologically older? And the way that people test this is to ask whether are people who appear older, if you follow them up for many years down the road, do they develop disease? Or are they at higher risk for mortality? And so we were able to test this in one of our cohorts, and we found that basically all of the organ age gaps were associated with increased mortality risk over 15 years. And so this is, the x-axis is a hazard ratio, so roughly percent increased risk. So the people with older hearts, that's roughly a 50% increased risk of mortality over 15 years, probably reflective of heart disease being the #1 killer in the US, but also many organs increasing risk for mortality. So this was encouraging. It suggested that we're measuring something related to the physiological aging process. But we wanted to go even further and determine whether measuring aging at organ resolution actually gives you organ resolution physiological information. So in other words, if you have an older heart, are you at increased risk for heart failure compared to if you're older on other organs? And we found that to be true as well. So in here, over here, people with older hearts had roughly 2.5 times increased of future heart failure. And this was not detected by really any of the other organ aging or any of the other organ age gaps, which really suggested that we should be measuring aging at organ resolution because it is telling us something different about someone's risk for organ-specific disease. And one of the great things about proteomics is that, and these models, is that the models can pinpoint which proteins are actually important in predicting age and associating with disease. And what we find is this protein MPPB is one of the top hits, and TNNT2. But interesting thing about MPPB is that it's a hormone that's released in response to heart damage. It's measured in the clinic to diagnose acute heart failure. But what we find here is that this protein also increases during the normal aging process before any heart failure and is actually predictive of future heart failure 15 years in the future. So it really suggests that we should start measuring proteins not to diagnose people, not to assess acute damage, but also wear and tear over the decades, and that this is actually linked with future disease outcomes. And all of these proteins here are expressed by cardiomyocytes. And another really interesting and exciting opportunity here is, can any of these proteins also be drug targets for heart disease and so on? So we're thinking about that as well. So this is all now unpublished work where we've expanded our study. So that initial study was roughly 5,000 people using the SomaScan proteomics platform, and now we've expanded to a cohort at the UK Biobank of roughly 50,000 people, 3,000 proteins on the Olink platform. It's an enormous sample, really tons of data, and ton of phenotypic information, disease follow-up on the UK Biobank, which allows us to ask several more interesting questions. So one of the really interesting discoveries here was that brain age is uniquely predictive of future Alzheimer's disease risk. And this was really actually quite specific to brain age. So none of the other aging models really predicted Alzheimer's disease and dementia. And so here, as I'm plotting the, you saw that earlier, those group of extreme agers, the people have especially aged in a single organ. And so in the red are people with an especially old brain. They have a 3.8 times increased risk of developing Alzheimer's over 15 years. And the people in the blue are people with actually younger organs. So they have a really youthful looking brain. And they're protected about 80%, 70% for Alzheimer's disease. And if you look at the proteins that explain brain aging, you find proteins across the cell types in the brain. People might notice neurofilament, which is, I think, mentioned earlier. It's a biomarker of axon degeneration. But that's actually goes up in the blood in neurodegeneration, but we see that it also increases during the normal aging process, detectable in blood from people aged 40. We also see GFAP, which is a reactive astrocyte marker, sort of astrocyte inflammation. Also many other proteins in the oligodendrocyte and OPC lineage, so the proteins that myelinate axons. Some interesting proteins around the extracellular matrix. So I think we're capturing many different aspects and biological processes of brain aging sort of all together using these statistical frameworks. So instead of measuring each protein one by one, we get a holistic understanding of someone's brain age, whatever that means. But this really is predictive of future neurodegeneration. One of the things we wanted to determine is that we couldn't really ask in the first study due to sample size was, how does the aging of one organ compare versus having 2 to 4 aged organs versus 5 to 6 aged organs in someone's disease risk or mortality? And so we asked that question here. Interesting thing, we found that an older brain is really quite predictive of increased mortality, but also having 2 to 4 aged organs, so the multi-organ 2 to 4 plus. Having 2 to 4 aged organs is bad. Having 5 to 7 is even worse. Having 8 is really not good. So that's, you can actually visualize this here. Roughly 60% of people with 8 or more aged organs dies within 15 years. What's really quite amazing, though, is that people with young immune systems and young brains seem to be protected from mortality globally. And people with both, so young brain and young immune systems, are even further protected. One interesting fact was that people with 2 to 4 young organs broadly, or 5 to 7 young organs, were actually not protected from mortality. So this is a little bit surprising, but thinking deeper about this, there's a difference between a protein being too high versus too low. I think the example that I think about is the markers of damage, so liver damage. They really increase in response to damage, but being on the lower end of the spectrum doesn't really mean you have any less damage than someone who's normal. So perhaps the other organ aging markers are more related to damage accumulation, and maybe the brain and immune aging markers are more related to function on a continuous scale. That's sort of how I think about it. I'd love to actually get your thoughts on that too. But it really suggests that the brain and the immune system, and having a youthful brain and immune system, is key to human longevity. And I think inflammaging immune system, that's really quite common. But I think, as we've heard from today, these autonomic functions and stress and depression, pain, all have such huge impacts on health, and perhaps that's actually reflected here, that what we're measuring in brain aging is not just neurodegeneration, but some global process related to various functions of the brain and communication between the brain and immune system too. If you look at the proteins and the pathways of brain and immune system aging and resilience, you actually see So for the brain, you see perineuronal net and synapse-associated extracellular matrix being a top pathway, which is quite interesting. So these are the extracellular components that wrap around neurons that actually support synapse function, and these are produced by oligodendrocyte precursor cells. Again, suggesting not just neurodegeneration, but some sort of maybe global brain ECM. And also, if you look at the top immune pathways, you see secretory granule perhaps related to cytokines. I thought neuroinflammatory response was quite interesting. And perhaps there's some relationship between these neuroinflammatory proteins and these extracellular matrix brain proteins, which we're starting to sort of explore computationally. Okay, organ aging, we also got to explore this in the UK Biobank. Tons of people, tons of phenotypes. It's also associated with lifestyle choices. So as you might expect, alcohol and smoking is pretty bad for you across the board. So is processed meat, we see here. And oily fish and vigorous exercise is associated with youth across many organs. So this really suggests that these aging markers are not just genetically determined, but they're also sensitive to the environment and our lifestyle choices, which is encouraging for us, but also encouraging in the sense that we can measure how we respond to environmental factors at an individual specific level and at organ resolution. We also were trying to understand whether certain drugs could be rejuvenating for certain organs. And so we looked in the database for people who, for drug intake, and we found that estrogen treatment, which are prescribed for postmenopausal symptoms, is associated with immune system youth. And this was quite interesting because it's been shown that early menopause is associated with many comorbidities and diseases. And actually, we see this reflected in the organ aging models as well. So in the reddish color, you can see the effect of menopause on organ age gaps. It is associated with increased organ age gaps across the board. But the women who take estrogen treatment actually have what we think may be a sign of immune youth, so reversal or protection from age acceleration, especially in the immune system. And perhaps this actually prevents or protects from mortality. It's been shown very recently, actually, in the UK Biobank that estrogen treatment is associated with reduced mortality risk. And so perhaps it's through rejuvenating the immune system, which we also showed earlier was protective. So I think there's lots of potential here to explore. First, monitor organ aging in living people. Everyone's different. But also, how do certain lifestyle interventions or drugs affect organ aging at the individual level? I think this is one of the key barriers to assessing a longevity intervention in a clinical trial is you have to wait a really long time to see if it works or not. But if we have that first clinical trial and we measure all of these different proteins and these different biomarkers and really find surrogates for something that is truly rejuvenating and protective, we can accelerate clinical trial development and start doing screens for rejuvenating molecules. We can also ask, do interventions rejuvenate the whole body or just specific organs? Are there accelerated aging side effects in all or a subset of people? This is also probably going to be really important for predicting safety of a drug. And also another interesting point for drug discovery, discovering new targets or new proteins. Can the proteins that we actually measure to monitor aging also be the targets to try and reverse the aging process? Cholesterol is a really nice example where we know that it's in the blood and it's high, and there are drugs that try and reduce cholesterol levels, and that's beneficial. Can we do the same with some of these new proteins, maybe in the brain, related to brain extracellular matrix or the immune system inflammation? And so with that's the perfect timing. That's the story of how the first step of trying to measure organ aging in humans to get a better grasp on health and disease. And I didn't have time today, but I also wanted to mention that we're using the same proteomics platform now in cerebrospinal fluid to try and better understand Alzheimer's disease. And we really find there's this really interesting signal of synaptic aging being a key component of Alzheimer's disease that interacts with the pathologies. And I think it's been very reported in studies, but not great evidence at the molecular levels in humans. And I think we're making big strides there. And with that, I'd like to acknowledge the lab. Many different people have contributed to this. There's so many different study cohorts that I did not generate any of the data. Collaborators, Anthony Wagner, Beth Mormino, all of these people basically leading the different cohorts that we've worked together with. And of course, SomaLogic and Olink and have been absolutely critical to all of this work and the funding sources. So thank you.
So who's got a mic?
I have a mic and a question from online.
Okay, so Larry, and then right here.
The question is it nature or nurture? It's both. Your study and hypothesis don't appear to include social determinants of health, and that might be a bias limiting your answers.
Yes.
Do you have thoughts about that?
Is it nature or nurture or both? This is an interesting question that I think about a lot. So that's the first one. I think it's mostly nature in the sense that our human lifespan is fairly capped at 120 years. No one has really lived beyond that, and even with extension of average lifespan of the human population with global wealth, we've never surpassed that cap. And also species live really dramatically different lifespans. So that part is nature. But then there's so much in the environment that explains heterogeneity and health and lifespan within our species, within humans. I mean, from dying within 40 years to 120 years. And that's definitely a meaningful difference. So I think there's lots to do with the environment, lots to do with all of these social determinants, chronic stress and depression that I think are harder to measure, and so the field has not gone in that direction, but definitely an important one to try and figure out the molecular biology and physiology of all those things. Yeah.
So Jim?
Hey, thanks. Great talk. I'm Jim McSwiggan. I had a question that could be for you or it could just be for SomaLogic, but I'm interested in the proteins in the blood. Do you envision this as being cells that are breaking open, dying, breaking open and releasing proteins, or are all proteins being excreted to some degree from all cells?
Yeah, it's a great point. I think it's not just cell death, because I mean neurofilament, which we typically think of as axonal damage, you can measure it in a 20-year-old, which suggests that it's a part of homeostasis. And unless we think that there's constant turnover of neurons, maybe which is true, I don't know to what degree. Yeah, I think it's probably more than just cell death. And there's actually lots of membrane proteins as well, some cell membrane proteins that are cleaved. And so there's that level of regulation of how proteins end up in the blood. There's probably secretion that we don't really understand, beyond the traditional sense of secretion. But yeah, there's a lot in there.
Okay, up here?
Yeah, my name is Ramesh Akina. I have a question. Women in general live longer than men. Do you have any comments on that?
It's true.
What determines it?
One thing we find actually is that men have older hearts, and heart disease being, the most common cause of death, I think is probably a big one. But I don't know. It's a good question that we haven't really thoroughly gone after because it's already so complicated already, but I think the next step is definitely really figuring out sex-specific patterns of aging. Not just are women younger or older, but what trajectories of aging only happen in women versus men. Like reproductive aging is a big one. Yeah.
Okay, back there, then over here.
Are there any plans to employ any interventions and then see if you can reverse, like the aging profiles of some of these organs?
Yeah, we'd love to do it. I think money has been the biggest limiting factor. Everyone wants to do it now. Like, how does exercise affect aging of organs? The more the samples are measured, the more we're going to find out it's going to happen in the future. Yeah.
Over here.
Yeah, my name is Ben Binder. Regarding lifestyles, I'm 79 years old, hardly any gray hair. I ski twice a week. I went to get a physical a couple weeks ago, and my primary care physician said I look pretty good. I said, well, doc, there's a couple things I do. I eat well, I get exercise, I rest, and I hang around with young women. And he smiled. Then I told him I'm 79, the last woman I spent the night with was only 26 years old. And he kind of looked at me disapprovingly. Then I said, that was 32 years ago. But it seems like just yesterday.
Okay, so Larry down here. Do you want to follow that one?
I actually do, but I don't. But I would like to ask you something you said in the very beginning that surprised me. So, as background, I thought I had read carefully every paper ever published since your lab started doing this, and I missed what you said, and I want to make sure I got it right. You suggested that you did the bigger study, the one that led to the Nature paper, which is my favorite paper of all time.
Really?
I'm serious. Watson and Crick would be second. But what I didn't understand when I read that paper, I thought carefully, that you started thinking you were going to find organ differences in aging, and I thought from reading the paper that it emerged from the data. And the question I'm asking you is to tell me what the truth is, and I will remember it that way forever.
Well, first, thank you for the kind words. That means a lot. So we didn't. I mean, we stumbled upon it in the sense that we did discover that having an older organ was actually associated with organ disease. But I think we did always hope for that to be the case. Yeah. And that really started from, I think, Tony's work in the mouse aging atlases, discovering that, aging really can't be explained by a single biological process, especially when you look at all of these different organs and how they change so dramatically and differently. And also by many other studies in the field that have suggested that organs age at different rates, even in flies. Like in flies, the skeletal muscle and the intestines seem to age first. But is that different in mice? Is that different in humans? So we were very much interested in trying to see what human aging really is like from the beginning.
Back here.
Richard Barker, one of the things I'm doing is creating a major foundation on healthy longevity and interventions. So we should probably talk with a simple curiosity question. The relationship between what you're measuring and the much more talked about epigenetic clocks.
Yeah, it's a good question. I'm biased, but I think the epigenetic clocks have an inherent limitation in that they only measure epigenetic marks on peripheral blood cells. There's no way you can ever access epigenetic marks from human brain tissue. I think that was one of the motivating factors early on, was that the plasma proteome lets you access basically any tissue in the body as long as that tissue secretes proteins and you can measure them. And And also the plasma proteome, I think, is much more dynamic, and there's known functions of proteins, and you can modulate proteins. The vast majority of drug targets are proteins. So there's also improved explainability. I think epigenetic clocks are more interesting in the context of evolution and of maximum lifespan. So actually the rate of epigenetic aging is really highly correlated with maximum lifespan of species across the mammalian kingdom. And so can we find sort of a rule to epigenetic cause or rule to aging that's evolutionarily conserved? I think that's where epigenetics has its use. I think proteomics is much more focused or much better at disentangling heterogeneity within our species, within humans, and health outcomes in particular. Yeah.
Okay, so we're back there, and then Richard after that. So back there, you with the mic, yeah, you're up.
Okay, I'm on. Jamie Haywood, and I'm going to echo Larry, but narrow the field slightly. I think this is, without question, the best proteomic paper ever published, and I think the work is magnificent. I think what I love is that you sort of added a layer of explainability to the complexity of machine learning to help people understand it, make it more accessible. My question is, because I do some similar work, is have you compared the prediction of sort of an organ aging-based clock to actually directly machine learning to the time to disease or the time to adverse event? Because they give different kinds of power and information. And I think there would be a lot of value, because I know the performance of the direct models are actually quite a bit better, but they're unexplainable. And so the ability to sort of look at the things in comparison in different ways together, I think, is really magnificent. And again, I just want to compliment you. Beautiful, beautiful work.
Thank you so much.
Go ahead.
I think it's a really important question, actually, because I think the field is trending towards making mortality predictors, as you mentioned. And one of the key limitations in my eyes is that how do you know whether you're measuring that's something that's aging, that's changing over time, or something that's just like an environmental stressor? Like if someone smokes, that probably does affect aging pathways, but it also is additive damage that might have nothing to do with aging. And so then if you have a model that's trained to predict that, then is it aging? And maybe that's more of an academic debate because actually even if it's not aging, it might be really important for health. So I think both angles are important and worth pursuing, and I think the key will be really disentangling what is aging versus environment or environmental stress, and then actually the interaction between the two. But yeah.
Richard?
Exciting presentation, and but if we look back over the last few decades, we had blood pressure screening, and then we had cholesterol screening, and with each of those, the actual methods, especially for cholesterol, became cheaper to measure, and we have different approaches now for different kinds of hypercholesterolemia or different types of high blood pressure. And then we get precision medicine, which comes with genomic analysis. And the cost of genomic analysis has gone way down. With what you're looking at now, do you think that maybe the costs will go down and that we should institute this approach, these approaches? I worked on cholesterol screening in kids. Should we be having 40-year-old aging screening? Or what age should we start doing this? We know Alzheimer's, for example, the pathobiology starts years before the clinics in most of the cases. How should we be applying this in the future?
Yeah, that's a great question. Well, the first one, I think costs will probably go down just because they seem to always have. Beyond that, I don't really know the economic details but in terms of employing them in the clinic, I think what we need to do is figure out actually how to treat someone if they have an old brain, because we can't do that right now. There's no treatments, and so it seems that one of the major drawbacks from having a biomarker that you measure is if you can't act on it, then why measure it? So I think, for the exploratory people that just want to know their age, that'll probably be an option. But in terms of integrating into clinical practice, I think we'll need to have a drug, like the cholesterol lowering drugs or like blood pressure lowering drugs. We'll need to have something that can actually intervene.
But 60 years ago, we didn't have cholesterol-lowering drugs, and I think that with these approaches, we're going to find drugs
Yes,
that
I
are going
think so
to be
too.
able to target many of these abnormal aging products.
Yeah, I think so.
So we're running out of time, but I'm going to ask two quick questions.
Sure.
So one, there are genetic mutations which drive disease. And I'm thinking of ApoE alleles, ApoE4.
Yeah.
In your biobank sample, are there enough people so you can tell, is ApoE driving Alzheimer's because it makes an old brain? Or is that a fundamentally different process?
Yeah, it's
That's
a
the
great
first
question.
question.
It's a great question. Basically, a question of like interactions between our genes and environment in aging and disease.
Well, are disease-causing genes driving aging, which
Yeah.
gives us disease, or are those separable?
I think it's probably a bit of both, and depends on the genetic variant. And I think a really interesting thing would be to figure out which ones do what.
And so
But yeah.
my last question, because we're going to have to move on, how do you think insurance companies would use this information?
I hope not
Yeah, okay.
to charge people, but I don't know. Sorry.
Well, let's thank Hamilton for a terrific talk.